Okay, the next topic is the stability topic. It's going to be led by Tona, who was introduced earlier on, so I won't repeat that. She is a member of the Quality Working Party. So over to you, Tona.
Thanks. And I'm very happy to introduce the speaker from industry who will give the presentation here. Ron Ogilvie. He is working in global CMC at Pfizer in Sandwich and is representing FBR. Please.
Thank you, Tona, for the introduction, and thanks to my colleagues on the working committee from Europe, from industry rather, because we went much beyond Europe in the end, to actually pull this presentation together. This isn't just a presentation from one company, it's a presentation that's got inputs from lots of different corners, and to be honest, it can never be fully comprehensive for a topic like accelerated stability. My colleague in Sandwich, Gary Scriven, said, "You need to take my book." Yeah, but then I saw how heavy the book was and I was going to Scotland, so I didn't bring his book. There's a reference to it today, but it's a lovely big tome about the understanding of scientific accelerated stability. So for me, that kind of is a, let's not call it a doorstep, but that's a step forwards in terms of how much prior knowledge there is for some of these topics. So I'd like to thank Drew from Vertex, Matt and Sandra from GSK, etc., as well as some of my own colleagues for putting together some of this material. I will say that the facts of the matter come from the companies, but I'll give you a disclaimer. Anything that sounds like a commentary or an opinion is mine, and I'll take the blame for that today. So if I put a little bit of a story around it, the story is coming from me rather than from the companies. But I think the story, in a sense, matters to this because accelerated development is about saving time. And one of the things about an impurity purge model is, yes, it saves time, but it's almost not real time, it's resource time. But a stability program takes real time to do. So actually speed in development can come from several different things. It can be getting to the initial clinical investigations as quickly as you can. It can be about managing the changes in development without those changes then slowing development down again. And stability can be key to that, as can some biopharmaceutics things. It can be getting to the registration submission as rapidly as possible. Now, I don't necessarily mean doing that with less than 12 months data, but that could be part of the overall story too. And of course, it's also about accelerated development only counts if it gets approved into the patients. So it's got to be done in a way that is acceptable to all significant parties. Of course, if you do something to try and accelerate your development and regulators don't think it's been an appropriate move, it won't accelerate anything. It will slow you down. So it's really important that this is done effectively, scientifically, transparently, etc. And I did keep the final bullet on the slide because in the regulatory discussion ahead of the meeting, I said, and it also matters for supporting post-approval change. And Tona said, well, why is that important for development? But actually, in the context of an accelerated development program, you're likely to be putting some matters that you might have done in development into the post-approval world. So again, if you want to find ways to facilitate acceleration, in my book, in my view, you also think about how to facilitate post-approval change. So for me, speed comes from all of these things. I've kind of talked a little bit about the second slide already. Obviously, stability, which takes time, is on the critical path for a development program that you're trying to do quickly. But of course, stability can be essential for managing and understanding product quality. And you'll have to do it to get to your first investigational study starts. You'll have to do it to develop and optimize a product through development. You have to have stability for registration, and you have to have it for many post-approval changes. So you see how intimate this topic is with the whole perspective from industry on how can we do things quickly. And I don't think it's just our perspective, because of course regulators want the clock not to get in the way of the patient delivery. The patients don't want the clock to get in the way. So we have to find some way to get around this idea of real-time data needs to be done for everything. There is in the Q guidances. Now remember, we've been talking recently about Q Q11, Q8, Q9. Their numbers mean that they were developed relatively recently. We've seen how strongly scientific the M7 guidance is. Q1 is of its time, and it was scientific, it was risk-based, it was thoughtful, but at the same time, it was maybe more conservative than this new textbook of acceleration understanding might need it to be. Because in the Q1 days, there was an allowance for alternative approaches and extrapolation, but those allowances, both in Q1 and even in the IMPD quality guidance from Europe, they're relatively constrained extrapolation allowances. You can do certain things, but there are barriers and thresholds to how much extrapolation you can have. It's perfectly reasonable. At the same time, science might say, yes, but maybe I can tell you more than that. So there can be scientific approaches to understand stability using accelerated conditions that can do things much faster than the time clock and get you information that is relevant to real-time storage. And it's also interesting that it can be done in a de novo sense and say this will be stable, or it can be done in a comparative sense and say this new product is going to be as stable as that previous product that you understand. So it can operate in two different ways to support rapid development and change management. Technical background. As I say, there's a whole textbook. The Keeg-Scrivens textbook. It's very new. Gary recommends it to me. It's on my desk for reading. I haven't read it all yet. So Gary gave me this slide that said, these are the basics, Ron. Please make sure you understand the basics. And the basics are is you can use accelerated stability studies to generate a mathematical predictive model that says, I can take the stability under accelerated conditions of a blend, of a drug substance, or of a product, and I can then tell you predictively what the long-term storage is going to be. I can do that by using accelerated aggressive conditions like 70-75. So clearly you're going to knock things out of the park in terms of stability challenge. But it's only appropriate to use those very accelerated conditions if the manifold of degradation stays the same. So you've got to be operating inside what you understand about the product. But if you can do that, you can take that very aggressive accelerated data and then predict it back using a kind of a modified Arrhenius approach that builds in both temperature and humidity, sensitivity, allows for factorization around both those, and you use an empirically derived logarithmic relationship between your degradant attribute and the temperature and humidity that you're using. You can also model in for things like packaging approaches. If packaging is important to you in terms of moisture uptake for degradation, you can model that around the packaging too. So in a sense, the accelerated stability approach is critical to time, but actually in the time since Q1 has developed a significant amount of new understanding. Now for me, that is a really important thing to kind of use as a background to the cases that I'll show you. I've got 4. They talk about different angles on how you can use accelerated stability. The first one was a case study where a company were developing a program. They had it well through development, but then they discovered a late-breaking stability issue. So they were getting to a stage where they didn't think that they would get a usable commercial shelf life under ambient storage conditions. And of course, then they're troubled, because how are we going to take that medicine through the accelerated development program, through the accelerated approval, and not then be able to deliver it to the patients? So to try and work through that then, they did several things. And it's really important to be able to do several things. And one of them is to be able to engage with your regulators who are going to understand and think about the package with you. And you have to have a regulatory system that allows you to manage these unexpected events. What happened first was they said, look, we've had this problem. We've got this stability data. It doesn't look like it's supportive of what we want to do going forwards. I need to change my storage condition. I'm going to offer you a commercial storage condition of a refrigerated product. It'll still be fine for the patient. Here's the data we've got on the product strengths under refrigerated storage conditions. Yeah, but they didn't have 3 batches of each of the product strengths. So immediately they're talking about less than a normal data set for approval of those products under that storage condition. However, working with the regulators, they shared the data they had, which was bracketing the middle strength. They had some data out to 12 months on the top strength and the bottom strength. And with the regulators, they agreed with that data set to apply for the top and bottom strengths, a 12-month shelf life, 3 months only for the mid-strength because they didn't have much data on it. But the regulators were working with them, and then during the review process and beyond, they could add additional data and extend the shelf life. Great, it's workable, it's workable. The other thing they were doing in the background was understanding what modelling data we might have. They got an agreement with the regulatory position in terms of how to support the refrigerated product, how to get their shelf life, how to manage it. It's quite onerous. It'll involve updates to shelf life, it'll involve updates to stability data, packaging, etc. The modelling approach would have allowed them to take that initial data set and model it back to refrigerated data, predict the shelf life under the refrigerated condition, apply that to all strengths which were, in a sense, QQF equivalent, and actually, in one fell swoop, using the magic of mathematical modelling and prediction, they could, in theory, have gone, I can tell you what that final data set will be. I won't have to wait for all that real-time data. I'll be able to give you the predicted shelf life now. Can we agree to that now on the basis of stability? Now, that's not how this was handled. It was handled in that very collaborative way through close engagement with regulators. That's a strong part of the accelerated development programme. And it got to a great outcome for the patients, but actually it could have been done in a different way and done more easily. So even when you get a great outcome from a collaboration, there's other science that can be brought to bear. I'll give you another example. It's important to get to the clinical development programme and move it quickly. Remember, in those early days, you don't know if you're a prime, breakthrough, adaptive, or a standard candidate with standard development time. So what can you do? Again, you've got the ability to do prediction and accelerated stability, but interestingly, it's not viewed with a huge amount of warmth in every regulatory arena. For example, if you look at the IMPDQ, you get the feeling that there's a kind of a reservation against using this kind of stability data, because it allows a fourfold extrapolation, which is great, based on accelerated data, up to 12 months, and it says other schemes may be possible but should be justified. Now, there is nothing wrong with that, and it assures the quality of the product to the patient. But in fact, strict enforcement of that kind of approach can mean that you need a certain amount of data to be able to get going. You need to then make updates as you do your clinical programme, etc. And actually, that IMPDQ guidance position is not the same in every country in the world. In some regions and countries, there's more flexibility, more acceptance of the science-based approach. And actually, that can mean that you can progress studies at different speeds in different regions, which is kind of interesting. Remember, you can't claim at this stage, this is my prime breakthrough candidate, because you're not even in the clinics yet. But can you move quickly? And again, the use of accelerated stability data can help you move quickly, compress the development times, and actually change your critical path. Here is one company's little bit of a history of this. Now, it's interesting that it goes back a little bit of time, because it predates things like the IMPDQ guidance coming into stream. But actually, if you look at the earliest one at the top of the slide, you might say that Country 3 back in 2014 were doing a really nice job supporting the use of accelerated stability data, because they gave a 24-month based expiry on the product based on that short acceleration data set. Now, since then, you see things have maybe tightened up. You can get less allowance in the later examples for your accelerated data than you had in the initial examples. Now, as I say, this is my spin on the story. It's not what the company told me to say. But it is interesting as you go through this, you get less expiry for the data you had originally, or you need further information, which is fine. Every company should always be prepared to answer questions for further information. But interestingly, what you don't tend to do is to get an immediate acceptance of an accelerated stability programme to establish the shelf life of your product. Most of these are investigational products. There's one at the bottom that was genuinely a breakthrough development program. It went to a non-EU country, and the final submission could only be made when 12-month stability was in place on the registration program. I'll just say that to you, and then think about that with the other examples that are coming up, because even that might be a little bit conservative in terms of what could be doable. The company was very clear that they wanted me to say that that was already a breakthrough therapy, a prime therapy candidate. Everything was in place to accelerate that, except we needed that 12-month stability data to go ahead. In all cases, the company made the commitment to study real-time stability data, and in all cases, it confirmed the predicted shelf life. There was no cases where they got a surprise. So again, that suggests that some of the science that the acceleration is based on is actually pretty strong stuff and worth considering. Another case study is about managing change. Development is change. Managing change is development. A compound here where the company had found that as they got close to registration, they could improve their API manufacturing process to improve the quality of the API, to change the process by putting reagents together in different ways, not changing the bond-forming chemistry, but by actually doing the chemistry different under a different set of controls, they found they didn't need the recrystallizations they were using. Great, makes the process cleaner and more efficient. Yes, but under the accelerated timeline, the original ICH program had been started with the previous process, with all those extra impurities in it and with additional recrystallizations in it. So now the company are saying, well, we can do better than that, but do we have to restart our ICH stability program? So again, transparency, discussion, and an allowance with the regulators to allow a comparison of the new process stability data with the old process stability data, and to use that under an accelerated condition to be able to compare one with the other. And then having got that comparison, using the comparison to say, well, the original data set is fine, supports our submission, and allows us to understand and set an expiry date. Again, a commitment to do follow-up testing. I'll show you by flicking forwards a little bit about what was done. The top of the slide talks about the changes to the final chemistries, changes to the processing, removal of crystallizations, and the fact that actually the impact here is to reduce impurities without changing the potential impurity profile. So it sounds like everything's heading in the right direction here. Things are getting cleaner. Stability was accepted on the basis of an accelerated comparison, done in 14 days rather than restarting the registration protocol. Really quite a strong case, I think. Now, it's interesting that that type of science doesn't just need to be done to manage a change which leads to improved quality. It can actually do things even more complicated than that. You can change a route, you can change the impurity profile, introduce new impurities, and still study the impact of stability change from that on the basis of accelerated data. So actually this acceleration approach can cover a large number of types of process change, provided you're understanding the degradation manifold and what's causing it. So you have to understand your program. It's not done blind, but it allows you to do things quickly. Of course, it can accelerate your stability program. It can accelerate development. This company found that they could optimize that chemistry without it becoming on the critical path to supply. And actually, that's fantastic as long as it plays out in every country in the same way. That's one of the interesting things about some of these stories. It doesn't always work out in the same way in different countries. So if I can find that one, back to case study 1. That agreement with the shelf life around what shelf life do I get for the refrigerated product based on the data set I've had. You can take that to one assessor in one country and get a single answer. You can get an agreement. If you take that same data set to other countries of the world, will you get the same answers on it? Will you get the same agreement and outcomes? Maybe not. But if you actually present the regulatory science of the accelerated conditions to all the regulators in the first pass, does that become an easier thing to globalize? So it's really interesting how the accelerated data can change not only your critical path, but your critical environment. And that's one of the things that industry is really interested in. In our regulatory pre-discussion, it was said, drug substance is much easier to predict than drug product. And yes, I would agree with you. But this last case is, again, managing a drug product change, because development is change. And you've got a product that has to change colours to differentiate the different packs, the different strengths in the commercial product that they didn't need in the clinical programme. You also want to get the packaging right for the commercial product. So you're making changes, but you don't want those changes necessarily to be on your critical path to registration. Now, yes, it can be more difficult to predict drug product changes under ASAP because things like physical chemical things are more difficult to predict. It's more difficult to predict, will this have an impact on dissolution? But actually, if you're doing it by comparison rather than by de novo prediction, things get a little bit easier. So in this instance, you had the clinical product that was essentially similar apart from colour and packaging, and you knew what its long-term stability was. You also knew how it behaved under accelerated conditions. So you could then take your pre-change product under accelerated conditions and compare it to your post-change product and allow that comparison to be made to see whether or not the changes impact stability or not, and to do that in very quick time. Again, by doing that, you can change your critical path to submitting the post-change product. Again, it can be done on a range of different types of changes. I think I've only got 1 or 2 slides left. So yeah, you understand, the principles is you have to understand your product before the change. You have to understand what degradation it might go through. You're thinking about its performance under real-time data set, which you've got, and how it behaves under accelerated. And then you can take the post-change products under the same accelerated conditions and do a simple comparison of the data set. And if you get less degradation or similar degradation post-change, then chances are you can predict that the shelf life and stability of the post-change product is unaffected. You could get real surprise. You could get some really rubbish stability post-change because the packaging doesn't work and you've made a mistake. Yeah, but it'll tell you very quickly that that's not a change you want to progress. So you'll stop that change and do something more sensible. Or you can be in the middle ground where in fact you get some difference in the stability performance of the post-change product, but that you can then think about what does that tell me about the real-term stability of the program? How different is the predicted shelf life? So I think there's a picture. Oh, it's build. I don't like build. There's a picture that says you have to understand certain things to be able to develop a data set of your pre-change product at real-time and accelerated condition, and then allow that to be done by comparison. And then the comparison becomes the important thing in terms of predicting the quality and performance of the post-change product versus the pre-change material. I'll show you a bit of data because the guys gave me some. Pre-change product down the left-hand side, and then 3 different strengths of the post-change product in a different colour. And immediately you see in, I think it was about 14 days, you see that 14 days under the accelerated 50, 60, 70 conditions, you're not seeing any detrimental effect on the stability of the product. So quite quickly you can say we're very strongly sure that this post-change product will not impact on the stability. And you can make that case then to say that my clinical data set under real-time conditions helps support the understanding of the stability of the proposed commercial product without putting that on the critical path to submission. I remember that story where we couldn't get the breakthrough therapy product approved until you had 12 months data set on it. Well, which of it needs here 12 months data? Is it the pre-change and clinical investigational product, or is it the post-change product that's on your critical path? And it's interesting because it becomes a discussion between scientists. And actually, if you want to make prime adaptive breakthrough work, it has to be a discussion between scientists. And on stability, you're in a really great place because the science is strong, it immediately impacts time, changes the rate of product development, allows you to manage late differences without them immediately impacting your critical path. So it's almost one of the most exciting things that development scientists can do for the wider clinical program is to say, I understand my product stability and quality without having to put it on real-time stability data. You can do that without affecting the product quality. You can still be very sure of the science that you're going to be developing on, and you can manage any attendant risks by doing it by comparison rather than by de novo prediction. This really, for me, is a piece of science whose time has come. And it's something that maybe if you were getting into, could we redesign Q1, there might be more opportunity for people to use processes like this as part of that kind of thinking. And I think that we as Pfizer will be trying to use it in post-approval change management in the future under PACMPs and Q12, because the science is right, and that for me matters. I'm going to stop. Thank you, Tona, for your encouragement and bullying to say the right things, which I hope I did.
Thanks.
She keeps me right. We worked together previously on a quality by design workshop, and I really appreciate how she makes sure I do the right things. So thank you.
Thank you for an excellent presentation, and yeah. And now I think I will introduce the panel, and after I've done that, we can start with some questions and comments for the presentation itself, and then we can go more deeply into some pre-identified issues. So I think most people here have already been introduced, but in addition to myself from the authorities, we have two representatives from the FDA, namely Scott Furness and Ramesh Sood. And from industry, in addition to Ron, we have Mohan Ganapati, who has already been introduced, and we have Drew Kosmishin, who's sitting there next to Ron. And he's a senior director in global regulatory affairs and at Vertex. His group is responsible for regulatory CMC strategy for projects in early phase through marketing application and post-approval. Prior to joining Global Reg Affairs, he spent over 20 years in various roles within analytical development. So I think I will start myself to just make a small comment to your presentation regarding the clinical trials requirements, because we recently revised the EU guideline for clinical trials and quality documentation. And I know that the FBR submitted a comment related to this to introduce the possibility to accelerated stability approaches, and it was not taken on board. I've just looked back in the public available discussion of the comments, and people preferred the ICH stability data and would like to see those, but also for early development phases. But I think that for products that are prime candidates, which we don't always know, of course, at the phase 1 stage. But I think the assessors should be open to consider such data for phase 1, and I will do what I can do to give that message to clinical trial assessors, and I will have an opportunity later this week.
So, Tona, that's really helpful. I remember those comments going in and being a little bit disappointed that there wasn't a more positive response to it. But at the same time, understanding that regulators are looking to guide everybody with a kind of general set of positions that can be taken. Accelerated stability is not an easy thing to describe like that, but you're right. If there can be an openness to consideration of it, that will make a big difference. It's unfortunate that you can't say, and of course it really matters here because this is an accelerated adaptive breakthrough therapy. You can't say that at the earliest of phases, but nonetheless you can show the science you've got, and it would be wonderful to be able to get that reviewed rather than to be told just bring me 12 months data or whatever you're told to bring.
So it would be interesting to know why the reluctance to use the accelerated data. Is it just unfamiliarity with it, or is there experience that accelerated data was used in the past and it wasn't confirmed with ICH data?
Well, I think it's most inexperience with it, and maybe it's also less suitable for some kind of issues than for others. So, but I will ask people to be a little flexible and open-minded when encountering it for things that could be prime candidates, at least.
Yeah, just to inform you that, but maybe you see that in ICH Q12, we have under marketed products, chapter 8.2, we have a similar approach in order to simplify all the post-approval changes we have, but this is, of course, in the light of post-approval changes. But the concept, at least, is there.
I agree. I mean, the regulatory landscape continues to change to allow for some of these science-based concepts to be included. I don't call them flexibilities, they're science-based concepts. And actually Q1 is simply of its time.
You want us to delete it from the...
Do I want you to delete
Q12?
it? No,
Okay.
don't do that. I don't think Mohab wants to change Q12, does he?
Hello, it's Dolores from the Quality Office. I have a question because I didn't see it mentioned in your presentation. So you are planning to submit this accelerated stability data, but are you planning to submit or proposing to submit any real-time data supporting like 3 months or something like that?
I would say yes to both of those if it's
Okay.
appropriate and necessary. But sometimes the answer could be no. If you have a strong accelerated predictive data set, then maybe that is sufficient to allow an assessment to be made. And it depends a little. And I don't want to say you never need real-time data, because that would be wrong. But sometimes you might not need real-time data, dependent on the circumstances of the proposal you're making.
Okay. And how would you validate your model?
How would I validate my model? So that equation that's on the board, that
Arrhenius,
modified Arrhenius position, in my mind is kind of a prior piece of knowledge now, prior art. So it's not validated per se for each program in front of you. It's a piece of science now that works based on long-term use of those kind of approaches.
Okay.
Now, there may be others who can add science to my answer, but that would be my answer.
Where I was coming from is because I saw your case study too, where you put the different cases and what was approved. I didn't see any information on the actual stability of the product. So perhaps some regions or some assessors were more restrictive because of the actual data submitted.
So let me ask Drew to answer that maybe.
Yes, so I can comment on when, for early development programs like phase 1, if we're filing ASAP data, our initial approach is to just file the ASAP data with a commitment to do the ICH studies on the clinical supplies. And so typically we're looking for a minimum shelf life of 12 months for a phase 1 study, and the ASAP in the products we've used it on, it's well over 12 months. It'll support 24 or greater. And then we have we have followed up with the ICH studies, and we have not to this date encountered a situation where the ASAP did not predict the ICH. So again, in early studies, no, the initial filing doesn't include any ICH data, but in our attempts to use it as we go later on, we do have ICH data in there. It just may not be the entire 12 months, and then the ASAP is supporting to extrapolate beyond 12 months.
Yes, and I would completely agree with that, and would also say that companies can then put the clinical product on normal real-time stability data to confirm it in line with the study as it proceeds. So you're not left without data, you're just generating the initial position differently.
Yes.
So Matt Popkin again. Just building on that. Again, to Ron's point, I agree that the science has moved past the point where they are, I guess it's product-specific models, but they are product-independent scientific principles, like any other scientific principles, which we in industry believe now are so well justified. And there's plenty of examples in the literature that the references that Ron's quoted, where you can see the real-time data and the modelled data juxtaposed for different scenarios. And I think that backs up. There's enough in the literature now that it should just be considered established data. So to turn the question around, I heard Tone's feedback that yes, there's a lack of familiarity perhaps with some assessors, and therefore that would lead to a reluctance necessarily to accept it without verifying data or commitments to data. But is there more to it than that? Are there blockers in the regulations that there's a non-compliance issue that's perceived, or is that simply not there and it's just a case of familiarity and precedence in terms of the amount of evidence? So do we believe there's any actual reason, any blockers in the regulations that need to be addressed with respect to this? And I'm talking in prime scenarios as well, but honestly, I think that's at some point that should be more broadly applicable.
Joost.
Yes, so I would like to add something to Tone's enthusiastic point of view. So I don't think it's just the stupidness of the assessors. There are different scenarios. And in the whole discussion, I missed that. Because there's one scenario in the clinical trial material. During the clinical trials, the applicant always have control of the patients and of everything. So this is another thing. Another scenario I can go along with you very, very well is if it comes to life cycle things. So you change synthesis, then your drug substance is pretty much the same. And then you do some accelerated study just to make sure that this will have no influence on stability. I can go with it without any studies. But I'm talking about my daily routine of getting a license for a product which will go on the market. And there is, and actually, or especially if you think of generics, they normally have very small amount of stability data. And the conservative approach is you have the stability data, you do the extrapolation as written in the European guidelines. I think in FDA it's different as well. And then you can do some conservative extrapolations. And after that, you do some variations to elongate your stability based on real-time data. And if it comes to 3 years, you are not allowed to extrapolate anymore, because with 3 years you can live. So this is a conservative approach. And I must tell you, it's not being open to new ideas, but knowing the regulatory background. Because have you ever imagined if you see, then you have some predictions based on accelerated data, you have a product which is on the market, big selling product, and then you get in real-time studies some problems. Then each agency, each person in charge has to think, is it necessary to do a recall or whatever? So this is lots of problems. And so therefore I would say, especially for products like generics, where we have 100 different products on the market, I would stick or prefer to stick to the concept. I'm absolutely open to clinical material. I'm absolutely open to the variations for just getting scientific background. And I have to think about this prime situation. This is something in between, and this is what we are talking about here. But I would say for a classic generic where there's no medical need at all, I do not see the need of opening a 5 years shelf life just on modeling or whatever. So therefore, I really would say we should differentiate between these things.
Yeah,
and not put them all together and say, let's talk about science, forget about formalism.
That.
Because it's a huge difference whether you should go for a marketing authorization or whether you should start a recall from the market.
Yeah.
These are two completely different scenarios.
I completely understand.
So just, I want to just make that you are aware of these things, maybe from the development point of view, it's another perspective.
And as a regulator, that's a very fair and reasonable position to take. And it's interesting that when we were putting the presentation together, I was wondering whether or not I should use my accelerated data to say, you need far less data to accept a registration. And actually, we didn't think that was the best thing to say. We believe that that might be an opportunity for an innovator, but actually the case that we made today was that the innovator would still provide you with real-time data, but it might be on a product that is comparable, i.e. the clinical development investigational product, rather than the final route of synthesis, rather than the final colour in the final pack. You would still get data to meet your needs, but done in a way that changes the critical path to getting the product to the patient. Now that doesn't, I don't think, set an unreasonable precedent for the generic entry market, and there may be ways to accommodate that kind of approach in regulatory thinking. Might not even need a change to guidance, because it's just this business of other approaches may be proposed if justified. Sorry.
Hi, Stuart here. Just to remind people that we aren't talking about stability only to support the approval. And I think I like Ron's use of the term that the dossier becomes a scientific discussion. With most of these things that we're talking about, we also have a GMP obligation around stability. And I think that's something that we should recognise, that irrespective of the nature of the scientific proposition we put forward in the dossier, underpinning that in some sense will be long-term stability looked at as part of our product review on an ongoing basis to ensure that that product is keeping to the covenants that we have set forth in the dossier. To me, that is the right place for that kind of stability.
Then it's Brian.
Thanks, Tony. It's Brian Dooley in the Quality Office in EMA. Could we see Ron slide number 7, please, Camilla? I just had a question on slide 7, Ron. This is in case study 2 where you had the 4 countries, and I'm just wondering, is this the same product and the same data set that's been submitted 6 times, or is it 6 products? Okay.
But coming from the same company, so my understanding would be that their approach would be similar, but it's been met with different responses from the regulators at different times.
Okay. Well, I suppose just to echo what Job said then, we've got 6 products, 6 data sets sent to 4 countries over 4 years. The inference earlier was that the extrapolation of plus 12 months is that one size fits all doesn't apply. But I understand. But in this case, you can see country 3 at the start has gone beyond that. And we don't know, I'm sure you've seen the particulars, but we don't know the particulars here. So we need to be careful about making comparisons here, because it seems that there could have been, product-specific assessments made in each case, which could be perfectly valid, and we don't know that. So I think it's a little bit, it's interesting, but I don't know if we can draw huge conclusions from this, because as Job says, there's context to every submission, every data set, and what type of information is in there. So just a note of caution on that one.
I think that's an excellent point, Brian, and I apologise if I gave the impression that this was bad assessment. I've got no evidence for that at all. It's just across time there looks to be some differences in the way that regulators might be responding. Now that could be for good and real reasons or not, or it could be because since the guidance came into play, regulators are less encouraged to be able to take that extension than they were before. I don't know which it is, and I should have presented both. So I appreciate your correction.
No, I mean, in some ways, with the EU system and how you submit clinical trials in multiple member states, if you had the same data set that was submitted in this, then you could maybe start to create an argument and look at how guidelines are being implemented and assessment is being done. So maybe that's something you could look at as industry. If you have the same data sets being submitted in multiple member states or regions, then you could actually, make an assessment.
I'm sure if there was interest from the regulators, we could bring back further information to kind of add flesh to this kind of initial slide. This was just one slide brought in from one of our companies as an example. You're right, it can be read in different ways.
Well, one more question.
Well, I guess it's a slightly different thing, taking it off a slightly different tangent, but one of the things that we've seen a lot around in the last couple of years in regards to stability has been this, particularly in some of the newer ICH companies, territories, has been this issue as well of repeating forced degradation studies. And I wondered if, Ron, you had any comment on that at all?
I'm tempted to say it's out of discussion scope for this meeting. You know, every country's got a right to look for the data that they want as part of their regulatory process. It's unreasonable for any company to tell them that they can't do that. Nonetheless, you would like to see some overall convergence of regulatory expectations that is science-based.
Yeah.
Now, if this is science-based, it would be wonderful. If there's others out there who think it's less than science-based, then maybe it could be done differently. That's as far as I'm going on that one today.
Well, I guess the reason I asked is because, I mean, there's an awful lot of talk about sort of prior knowledge and science, and I guess my sort of thought on, as far as forced degradation is concerned, forced degradation basically tells you what the primary degradative pathways are. They're fundamentals of the molecule, and it seems somewhat strange that then, just because of minor changes that you make, like a reformulation or a new API supply, that you'd be asked to do a forced degradation study, because scientifically it doesn't make sense. That molecule fundamentally hasn't changed. It's the same carbon-carbon, carbon-oxygen, carbon-nitrogen bond. So I guess I'm raising it a little bit to make a point that I think this is an area where we've seen a lot of creep recently, particularly in some of the newer ICH territories, and it's slightly concerning because it doesn't seem to be done on a scientific basis at the moment.
Well, thanks a lot to everybody who contributed, especially Ron.
Thanks, Tona.
This was definitely a team-based thing, and I didn't mention the people who were on the team really at the end. I forgot to put that slide up. Sandra,
Before we finish, can we just pick up on the physical tests bit? The physical tests aspect of this, we've obviously done the sort of the chemical stability. What are the particular challenges with physical tests, or have you got any experience of difficulties with?
Yeah, I can comment a little bit on that. And I think one of the issues is, do the physical tests follow Arrhenius kinetics like degradants do? The answer is I don't know. I do know that in our development programs, we did encounter a drug product that on regular ICH stability data showed a dissolution slowdown. So we decided we would investigate that using the ASAP approach. And we were able to show that using the ASAP approach, we could predict the dissolution slowdown that was confirmed with the ICH studies, and we got good agreement with that. Again, that's one example. I don't know if every example would show the same. But part of our use of ASAP is through all the accelerated conditions we look at, we do all stability indicating tests. So it's not that we ignore all the other data. If we were evaluating a new product and we saw dissolution slowdown through the ASAP study, we'd do some further investigations, try to understand that before we would go wholeheartedly and say ASAP can support our expiration.
Thank you.
Okay, so what you're saying is that you do all the physical tests under the ASAP conditions,
Yes.
in addition to the
That is correct.
to the chemical degradation and impurities. Okay.
That is correct.
Okay. Good.
So just let me add my thanks to those who contributed. Drew clearly contributed. My GSK colleagues, Matt and Sandra, contributed. Gary Scrivens from our Stability Council contributed. But there's a huge number of scientists who've now contributed on accelerated stability. I just got the pleasure of being the mouthpiece today. Drew said I needed to put in more data, and of course he was right, because he's just answered the question about it. Thank you.
Okay, thanks to everybody for their contribution. There is now a coffee break, and we're due back in the main room at 3:30, so thank you.